This edition of The World Today will examine the prospects of a next pandemic and the role of animals in transmitting to humans diseases like bird flu, swine flu, COVID, and other diseases. As climate change and human encroachment on animal habitats bring humans and animals into even closer contact, the risk of infectious pathogens jumping from animals to humans, known as zoonotics disease, will grow. So we're really happy to bring to you today an expert panel who can help understand this phenomenon, and we hope give us some advice on preventing future epidemics. I'd like to introduce our three guests. Andrew Hoffman serves as the 13th Dean of the University of Pennsylvania School of Veterinary Medicine, an acclaimed researcher, clinician, teacher, and mentor. He's a large animal veterinarian who has devoted his career to clinical service and research focused on lung disease, including new therapies for emphysema and asthma. He's chair of the Climate Change Task Force for the American Association of Veterinary Medical Colleges, exploring climate change and health education. Recently, he attended COP27 as part of the Penn delegation. And for all of you both online and here in person, there are longer bios online. Louise Moncla is an assistant professor of pathobiology at Penn School of Veterinary Medicine. Her research draws on tools from phylodynamics, virology, and population genetics to understand how viruses evolve with individuals, between populations, and across continents. The ultimate goal of her work is to better understand viral evolution and transmission to prevent new outbreaks from occurring and to mitigate the toll of endemic viral transmission. Last but not least, we are very, very excited to welcome our moderator, Benjamin Muller, who is a health reporter for the New York Times. He has reported on COVID with a focus on vaccine development, the mortality impacts of the pandemic, and the origins of the virus. He was previously a correspondent for the Times in Britain, for which we forgive him, where he covered the path of the virus in Europe, the evolution of the coronavirus, and related global vaccine distribution efforts. Thank you all thanks to our guests for being with us today, and I welcome them to the stage.
Thank you all so much for being here, and to the Perry World House for convening what is such a timely discussion, really. So we're here to talk about animal viruses and how they become human viruses. There are at least 10,000 viruses that we know have the capacity to infect humans, but the vast majority of them are sort of content to circulate silently in wild animals. And our question today is why some of those pathogens ping across the species barrier and come to infect humans. What sort of creates the conditions for those spillover events? Why is the threat of those spillover events growing in some ways? What are the consequences for animal health and for human health? And what can we as sort of scientists and citizens and policymakers do to try to address some of those risks? So I think in the introductory remarks, we talked about climate change forming the backdrop against which disease risk is changing in so many ways. And so I wanted to ask you, Dr. Hoffman, why the warming of the planet has changed the conditions in which viruses can cross from animals to humans?
Yeah, that's a very good question. And I think if you look at the emergence of pathogens from animals to humans, what we call zoonosis, we know that there are a number of barriers that the pathogens have to cross. And importantly, when you think about it, humans have to be in proximity with the pathogen. Those pathogens, whether they're parasites or viruses or bacteria or other organisms, have a life cycle. Some of the life cycle is on the host, lives on the host. And then the host also might be just a reservoir. So it may be a reservoir like Hendra virus in the horse. It doesn't cause much disease, but when the human gets in contact with the horse, with the disease, there's a very high fatality. So there is the pathogen who has its own life cycle, which may be even in water where there's poor sanitation. There might be large amounts of parasite eggs, for example. The life cycle is impacted by changes in temperature. So both latitudinally northward and altitudinally changes in climate can influence the range at which parasites or other pathogens can survive either on the host or the reservoir or in the environment. So you can see right away the ecosystem where the animal host, the reservoirs, and the pathogens live have many stages that can be influenced by climate. So heat, drought, extreme weather events, of course, flooding, standing water with heat. That's the worst thing for engendering mosquito larvae growth. The entire ecosystem by which zoonotic diseases exist is influenced by climate. So when we talk about climate change, we're talking about global warming. So that's also on land, on the surface. It's also ocean, as well as ocean acidification. And the other big areas are the drivers of that influence biodiversity loss that are also implicated, from which climate is implicated. So deforestation, habitat destruction, and extraction of natural materials ultimately reduces the biodiversity richness, and biodiversity richness is inversely related to the ability of pathogens to cross from either a free living state or some sort of a free living animal to the host of the reservoir. So you get it, there's a lot of different issues that interrelate: climate, biodiversity, and of course, poverty, which encourages fragmentation of the environment and extraction of natural resources and migration of people into new forests and so on and so forth. So I think climate is intimately involved in that entire cycle.
I think that one of the examples you mentioned there is a really telling one, the Hendra virus, which I think originates in fruit bats. And there's been some really interesting work about fruit bats losing some of their sources of food, in part because of warming. They feed on nectar from eucalyptus trees. Those trees stop flowering quite as much when the ocean's warm. And at the same time, because of deforestation, you've got those bats moving into closer proximity with farmed animals like horses. And that's sort of, I think, a good example of the ways different factors interact in ways that put humans into closer proximity with those viruses.
That's a perfect example. And Raina Plowright from Cornell has done a number of studies in Australia looking at this. I'll let Louise also add to this, but it's a flying fox is really, it is type of bat, is a flying fox. And what's important is that the natural ecosystem of the flying fox relies on regular nourishment from eucalyptus flowers. During El Niños, which normally happen every 5 years, but climate is driving to be in more accelerated and even greater intensity. During those disruptions, what happens is the eucalyptus flowers die, and the amount of food that's available is more limited. Those flying foxes move into agriculture and urban areas. And so again, it's another theme, that interface between nature and the agriculture and urban environment. As they congregate in these clusters, and these clusters can be tens of thousands of these flying foxes, and they're rather large animals. As they cluster, they compete for food and they exchange pathogens, including the Hendra virus. As they get closer to agricultural areas where horses are grazing, the horses pick up the virus in its sort of free living form and from bat droppings. And then, of course, humans come in contact with a horse with symptoms. So it's a really good example of how climate and climatic events can influence the behavior, in this case, the congregation behavior and the parsimonious behavior of bats. And bats are the number one, probably the species of greatest concern with respect to zoonosis presently.
Dr. Monkley, you study both the factors that sort of contribute to create opportunities for viral spillover events, and you've also done some really interesting work lately on the sort of way humans shape the environment in which those viruses then spread person to person. So you did a really interesting study contributed to a really interesting study using sort of genomic analytical methods to look at how the coronavirus had spread in Seattle and how it affected different parts of the county differently depending on structural factors like healthcare access. I wonder if you can talk a little bit about the ways that we're also shaping the conditions in which animal viruses spread among people.
Yeah, absolutely. I think in thinking about novel virus emergence, there's sort of two phases. There's this phase where the virus has to be able to get into a new population and start transmitting among people. And then once we have an endemic human respiratory virus, a virus that's really good at spreading person to person, all of these ways that we interact with each other can really influence how those viruses are spreading and moving and differentially impacting different populations. And this likely depends on some combination of all of these complicated factors like immune status and how we interact with each other and how we move across the world. And so this is kind of a broader theme in thinking about viral transmission in endemic viruses. You know, we know that seasonal influenza viruses spread really rapidly across the globe every year. So if you sample viruses in Philadelphia and sample them in in Europe, you're going to sample about the same viruses. And that's because we as humans move a lot through airline travel. And so we can spread these pathogens really rapidly. I think one thing that's been really interesting and also a little bit disheartening about the COVID-19 pandemic has, of course, been this renewed interest in health disparities and the role that these different access to different resources really differentially impacts COVID outcomes and transmission efficiencies. And it turns out this is likely true across a range of different respiratory virus pathogens. So, risk is not necessarily equal across the entire population. And this has to do with our ability to seek healthcare or take time off work, get access to vaccines and things like that.
Sticking with the COVID pandemic for a minute, I mean, while it may not be entirely clear how the pandemic started, we've learned a lot about the risk of wild animal markets from the pandemic, the potential that those were implicated in the start of the spread of the virus. And you, Dr. Munkle, have done some work, I think, looking at samples from Cambodian wild bird markets. And I wondered if you could sort of describe these wild animal markets are just such potent sources of viral transmission among animals and then from animals to humans. I wonder if you can describe what these markets are like and why they play an important role in zoonotic transmission potentially.
Yeah, so in full disclosure, I've never actually been to one. I've just read about them and interviewed people about them. But if we think about what these wild or these animal markets are, they're sort of the perfect place to observe cross-species transmission. So the area that I have the most familiarity with is in Cambodia. I have some really wonderful collaborators there who do surveillance for mostly avian influenza viruses in Cambodia and these live bird markets. And what makes these markets unique is they bring in all of these different animal species that wouldn't normally cohabitate together. So these are wild animals that may not have overlapping habitat ranges. And these wild animals will sometimes be kept in areas in close proximity to agricultural animals. So for example, like chickens or pigs. So it's all these animals that would never normally interact with each other. And suddenly they are. So if one of those animals is infected with something new, there's suddenly this new opportunity for that virus to spread to a new host species that it never had the opportunity before we brought it into proximity. And then because these animal markets are run and administered and they're for humans, of course, we interact with those markets as well. So for example, one of the best examples of this is that when humans are infected with avian influenza viruses, by far the strongest risk factor is whether or not you're a poultry worker. So if you do serosurveys of these people who work at live bird markets, a lot of them have, or not a lot of them, but a much higher percentage have antibodies against these viruses than if we were to survey the people in this room. So we're bringing in all of these different animals and then humans that can interact very closely with these viruses that can then kind of spread in this one location very well.
You mentioned avian influenza, and that's a big part of what makes this panel so timely. We're in the middle of the biggest bird flu outbreak in the history of this country. I think somewhere like 60 million farm birds have been affected by it. And this is a version of the bird flu that started, I think, in 1996 on a commercial goose farm in China. And so what I want to ask you, Dr. Hoffman, about is about what sort of impact this outbreak has had on commercial farms in the United States, on poultry farms, and the ways in which those farms have changed and really grown, especially the poultry industry in recent decades, and why that has also contributed, has also sort of driven up the risk of zoonotic transmission.
It really does relate to major consolidation and growth in poultry markets, and this includes turkeys, mainly chickens, but also ducks. Part of it has to do with our appetite for poultry has grown immensely. And this includes both the domestic market and a large market that we export to. So consolidation, growth in the size of operations, and then, of course, a larger export market. Some of the exporters, it's rarely ever live birds, but most of it is bird parts, and parts that we don't necessarily consume or wish to consume, the average consumer in the United States would want. So the growth of the export market, we also have maintained a biosecurity method for protocols to keep the birds safe from infectious disease, from the entry of pathogens into flocks as well as their transmission. And we do that through biosecurity protocols. And by doing so, by having very strict biosecurity protocols, and this goes for both poultry as well as swine, we reduce the chances of the introduction and transmission of the virus and ultimately zoonoses. However, and so there's a national poultry improvement program that monitors and sets the criteria and monitors all of these biosecurity methods. And if you join, let's say in the state of Pennsylvania, if you join and agree to, partake in this program and you fulfill these biosecurity protocols, you're also eligible for indemnity payments should you lose the birds. It's kind of an insurance policy, right? So if you don't sign up for the biosecurity protocols, which it's voluntary, then you won't receive the payment. So there's every incentive in the world to do that because it's very costly to lose a flock. Now, another issue, so the size of the flocks have grown, the markets have grown. We have both organic and non-organic markets that have grown locally. And they're also at the same time, more rapid growth, larger numbers of animals, mixtures of eggs, broilers that are used for meat, which grow up to be full market size in about 6-7 weeks. So very rapid growth, the genetics have changed. And then we have an egg laying business. In Pennsylvania, as you know, we have a giant egg business, not only for human consumption, but also for the production, ironically, of vaccines, which has been challenging. So you wonder why we have $9 eggs, right? So I think the reason, I think your question related to, so what are the dynamics, the changes in the industry that have led to greater risk? And I think it's got a lot to do with the size of the operations and the need for tighter and tighter biosecurities. The other thing is because we use the stamping out, in quotes, protocols to eliminate and to reduce the chance of the introduction of viruses into herds, rather than other methods like vaccination, right? We're always at risk. So if there's surface water that's not being filtered out and wild birds, waterfowl, have introduced the virus into that water supply, that's going to enter into the farm. And so none of these biosecurity protocols are perfect. Otherwise, we wouldn't have the original introductions of this particular latest strain into the commercial flock business. So biosecurity protocols are limited. And so you're all wondering, well, why don't we just vaccinate every bird? And that's because right now, the USDA uses the stamping out method to control infections in birds across the United States. And the import countries that would buy our birds will not take the birds because they haven't approved the vaccine. So we can get into the complexities of that later, but we're really relying on biosecurity, and the biosecurity methods are a little bit leaky. And then Louise is actually the actual expert on avian influenza and influenzas in general and viruses, and probably could state much more eloquently than I. Maybe you have additional reasons why we're losing the ground here, or have lost the ground here.
Yeah, sure. I mean, you certainly know more about the poultry industry than I do. That was like a great recap of all the ways the industry has changed. On the virus side, I would say that there's a lot of questions that we don't have answers for right now. this is the largest outbreak we've ever had in North America. We had a large outbreak of highly pathogenic avian influenza back in like 2015. And the reason this outbreak is so different from that one is that during that outbreak, the USDA deployed the stamping out protocol, which essentially means that if you find a positive detection on your farm, you cull all the birds on that farm. During this 2015 outbreak, a large number of birds were culled and then transmission stopped. So the virus went away. It was successful. What's different about this outbreak is a couple of key things. One is that almost 60 million domestic birds have been culled as part of this outbreak, and detections haven't stopped. So it's been over a year that we've had ongoing recurrent detections in both domestic and in wild birds in North America. The other thing that's quite different about this outbreak is we've had a lot more fatalities in wild bird species. So it appears that this virus seems to be much better at spreading and transmitting among a wide variety of wild bird hosts. The virologic reasons for that are not entirely clear. So this is an area of study that needs to be really sorted out, but this likely has something, this likely is really important for allowing these viruses to continue to be transmitted despite, in some cases, really good biosecurity protocols being implemented.
And one other thing, sorry, Ben, one other thing I'll add is that we do have backyard birds. The backyard bird flocks have increased in size. The live market, we do have live bird markets in Pennsylvania. They have grown in size. They're currently under quarantine, heavy restrictions right now. So you take a combination of wild bird incursion, live bird markets that are moving birds around, the trading of birds, for example, ducks across counties, not necessarily above ground, and the large flocks and the large in and out, constant stocking and unstocking of large farms and people coming in and out of those farms. The challenges are complex.
I guess the big question on a lot of people's minds with the bird flu now is sort of what it would take for this virus to adapt itself to people. We've had a few human cases of the virus, but it's not yet spreading from person to person very effectively, which sort of reduces the risk of a pandemic. I know, Dr. Monkley, you've looked at some of these specific human adapting mutations in samples of the virus from other countries. And I wonder what you've learned from that research about sort of what risks those mutations pose of making the virus transmissible among people, and what about the H5N1 virus sort of mitigates against some of those human adaptations?
Yeah, one piece of really good news is that we've had a ton of transmission in birds in this outbreak and really hardly any human infections. And that is really good evidence that there's some pretty strong barrier to humans getting infected and for this virus to begin transmitting among people. The barriers to cross-species transmission for these avian viruses into humans are complex and many and poorly understood. We know that there are a lot of interactions that the virus has to overcome with its particular host cell that it infects that's specific and different between birds and people. So it has to bind a different receptor in humans than in birds. It's actually a different temperature of replication in humans than in birds. So this virus replicates in avian intestines, which are much hotter than our upper respiratory tracts. So that's an important adaptation. And then humans and birds are quite evolutionarily distinct. And so there's all these different intrinsic immune factors that we have and that birds have that are really different. And for the virus to really successfully infect a new species, it has to overcome, a way to interact with our host factors and mediate our immune responses. So I would say that our understanding of what exactly those factors are and which mutations would mediate those interactions is very much incomplete. There have been shockingly few studies that have taken a really systematic approach to trying to define and understand those mutations. But that's something that I think the field is really excited about moving forward. And I think I'll stop there.
I want to sort of pick up on some of what we were starting to talk about the ways we're responding to the bird flu outbreak, our preparedness for a sort of broader transmission event. And you talked, Dr. Hoffman, about a few sort of prongs of a strategy like that. I wonder if you can describe sort of like specific steps on farms that we could take to reduce the sort of interface between wild birds and farm birds, if that's worthwhile. And you mentioned that the sort of idea of the vaccinating poultry at a large scale. Like, what do you think that's a strategy we should be pursuing more aggressively? How do you think about the industry trade-offs and the sort of health of the animal trade-offs there?
On the farm, well, let's let's start with the fact that some very large operations have never had any bird flu on their farms, right? So, and they're in areas where there are surrounding areas of risk. So there are breaches of biosecurity occurring all the time. So how the orders from the state veterinarians and the officials are pleading with farmers to constantly tighten up their biosecurity. Okay, and there's regular inspections, and as you see, because of indemnity programs, there's regular officers coming in, and of course, because of the National Poultry Improvement Program, there's regular testing, regular sampling going on all the time. So there's people providing external advice, and so, but we do have to improve our biosecurity protocols. But what we're seeing is, and Louise has demonstrated some fabulous data that she showed the other day, that there's movement of the virus back and forth between wildlife and birds. So it's a bidirectional problem, right? So there's leakiness in that system. And I think it's unrealistic to think there's ever going to be a perfect biosecurity system. Now, there are some innovations that we could introduce into farms to address that. Things like more effective use of disinfectant protocols. And it's costly because if you have to disinfect, you have to hire workers, you have to use the cleaners, you have to time washout, and so on and so forth. UV light kills viruses very effectively, and it doesn't kill the bird. So there are more effective ways. UV light's used widely for disinfecting water. It's used widely disinfecting sewage, and it's used widely, for example, on swine farms to reduce viral transmission and introduction. Very hard to do with birds because birds are in every little corner in these farms. But again, there's innovations there. There's innovations in the way we could be detecting the virus more rapidly. For example, wastewater. We've been advocating for wastewater. It's not part of the biosecurity protocol. We all know wastewater testing now. That's not something we would have been as familiar with before COVID, but we all know that that's in the municipality and in schools and even in dormitories at Penn. The effluent is tested. Yes, it was tested during COVID to get sort of mass screening at a very sensitive level. So we can introduce wastewater methods on farms and large operations to try to improve that. We could also, because in the case of avian influenza, it is transmitted largely between birds in fecal, saliva, this type of thing. Also aerosols, but not to the extent in humans. So there are better biosecurity protocols. There are better and more frequent and more tight diagnostic and surveillance methods. You have to have monitoring, you have to have ability to detect a virus, and then you have to have a very rapid response. So on the response end, we have a leaky system as well, because not everybody's, taking part in the perfect biosecurity protocols. And we also have, in some cases, bad actors. And we learned that during this outbreak. It was in no way the reason why this was such a vast outbreak, but it contributed to some of the confusion and some of the control and some of the testing issues. You asked about vaccination. We want to work with industry, okay? But at a certain point when and I'd be interested also in Louise's comments on this, but in talking with Louise this fall, the conversation went from this is going from an epidemic to, is it now endemic? And the word endemic has come up much more, meaning that it's here to stay. And do we know that or not? It seems as though the evidence is in the wild bird population, as Louise just said, it's becoming, at least the latest strains, is becoming more endemic. It's not burning out, right? Once a virus becomes, or a pathogen becomes endemic, that's the time usually when vaccination is introduced. So there's been a lot of confusion. There's the tension between pandemic preparedness on one hand, and on the other hand, the export market, as I mentioned, which is a major part of the revenues for these farms. So the idea of introducing a vaccine is costly, but there is also the element of pandemic preparedness. And so making that decision is difficult. We are advocating for vaccination, but a perfect vaccination hasn't necessarily been produced. There are vaccinations that are being used in different parts of the world, including Mexico and parts of Europe and South Asia. There are different types of vaccines. Our own Scott Hendley from Department of Microbiology at Penn Med has made an mRNA vaccine that has been protective in, well, so far has shown that it's highly stimulates a great immune response, and now that is in challenge studies in poultry. And so I think there's all likelihood that even Penn will produce a vaccine amongst the other vaccines, and that if we can get it deployed, we will. Now, Louise has also made a couple of comments to me throughout this whole thing, which I think is interesting. Poultry workers are more prone to transmission, right? So why not vaccinate poultry workers? You've made that point, I think, several times. So why not target your vaccine at a minimum for the humans that are at most at risk, right? Like we do, veterinarians all get, rabies vaccines. The other issue is, do we target certain populations of birds that are more at risk? The backyard birds, the live bird markets, and the genetic stock that we all rely on for future breeding of the birds, the highly valued genetic stock. We lose that. We lose our industry. So I think there's a targeted way to do that in both people and animals and tighten this all up, but we're not quite there yet.
I want to get to your questions soon. So start thinking about questions you might have for Dr. Hoffman and Moncla. But you mentioned, Dr. Hoffman, the importance of monitoring for these viruses, the idea that we don't really know what's spreading in wild birds, much less even in farm birds. And a lot of your work, Dr. Moncla, has sort of relied on those samples. We've been kind of blessed during the COVID pandemic to have so many viral samples that give you and your colleagues sort of this huge, rich store of genetic information that can help you learn about how the virus is evolving and spreading. I'm curious if you feel like you have the information you need to be tracking the bird flu, and also like the work you're doing, I understand, locally in Pennsylvania. I mean, I know the veterinary school here is involved in some of the diagnostic work for bird flu. What more can we do locally? You do locally to sort of build a better picture of what's happening in Pennsylvania, for example.
Yeah, so to answer the first part of your question, of course, nothing is as well sampled as SARS-CoV-2. I think one of the real bright spots of the pandemic has been that there are millions of SARS-CoV-2 genomes that have been deposited publicly that anyone can go use and analyze. And that's honestly incredible. We've had this real explosion in this scientific area, which is really, really exciting. So of course, compared to SARS-CoV-2, absolutely not. We don't have that many avian influenza genomes. I will say that there's a lot of people scattered about who are doing really good work on avian influenza surveillance. The problem with avian influenza that makes it really challenging to understand is that it's a global problem and that our surveillance systems are quite patchy. So there's really good data from some areas and really like no data from other areas. And that makes it really, really a problem. In terms of what we can understand locally, this really touches on something that Andy brought up, which is that one of our goals is to really try to understand which populations are truly driving transmission so that we can formulate vaccine schedules and strategies that really make sense, that are economically make sense. So one of the things that we're doing here is that a lot of these avian influenza diagnostic samples for birds that are suspected to be positive for highly pathogenic avian influenza come through the Penn lab. So we just, it's something really simple. We just said, do we have approval to start saving some of the viral genomes from these samples so that we can sequence them and then analyze them? So our plans for that data is to generate some more data, deposit it publicly so that other people can also use it, and then to use those genomes to try to reconstruct the transmission histories of this outbreak. And specifically, the questions we're going to be trying to answer is, how are these viruses getting into Pennsylvania and then transmitting within Pennsylvania? Are these viruses coming from repeated introductions from wild birds into these poultry systems, or is it spreading really well between poultry production facilities in Pennsylvania? The reason those questions matter is that they should really influence how we respond to this outbreak. If we're seeing tons and tons of introductions from wild birds, then we really need to make sure that we're keeping these domestic birds separate from wild birds and preventing those introduction opportunities. If there's tons of transmission between poultry production facilities, we need to think about how we're moving birds around to try to prevent that transmission from happening. So those are some of the things that we're excited about moving forward.
If anybody has any questions, maybe you feel free to start making your way to the microphone to asking. We have one question here from the Zoom chat that maybe relates to something you were just touching on, Dr. Moncla, which is about sort of how we respond and sort of reshape the structures in which we sell wildlife and you eat birds and so the question is about sort of what the average person can do to slow the spread of the bird flu, if anything, stopping the use of backyard bird feeders, for example. I'm also interested more broadly in whether we think there needs to be a broader rethinking of the sort of way we farm and sell livestock like poultry. I mean, we've seen during the COVID pandemic, mink farms be outlawed in certain European countries because the decision was made that they're just not worth the risk, given the way that virus was spreading through mink farms and potentially accumulating mutations. I wonder if either of you have thoughts on sort of how the average person can think about the sort of broader social response to this outbreak.
Yeah, for the average person, I would say I don't have a ton of concrete advice besides if you see a dead bird, probably don't interact with it. That's not a good move. In terms of our poultry or our agricultural systems, I think, if we want to be really serious about stopping zoonosis, we should rethink how we're interacting with animals and bringing new animal species together in a way that would promote transmission. Of course, this is a hard problem to solve because we have agricultural systems and these food systems for a reason, because they serve our needs for nutrition and food. So I don't have the answer for what we should do about that. But I will say that I am also pro reducing the number of mink farms, because mink also were recently infected with avian influenza. And there was this enormous outbreak on this Spanish mink farm where these viruses were transmitting among the mink. So it seems that the mink are weirdly susceptible to a lot of our viruses, and we would probably all be better served if there were fewer mink farms. But I'll let Andy talk more about that.
I'll just add that the birds that are most susceptible seem to be transmitting the most viruses are waterfowl. It may be because of their habits of transmitting the virus across the surface of water, and their susceptibility, inherently, the species are susceptible to virus replication. So not much we can do with bird feeders to make a big difference, honestly. But I do think that everybody who is in the proximity of waterfowl, and of course I work with the Philadelphia Zoo as well, and they've had to bring their birds in because many of them are waterfowl. They have them out now. They have the flamingos out today, actually. But you have to be very careful about the type of birds you are in proximity with, that you keep domestic birds away from waterfowl, for example. So there's some things that I think the CDC has written up very nicely in their website and that you can look at, but there's not a lot we can do in terms of changing our behavior, unlike some other dilemmas that we have in the world.
Please go ahead.
Thank you. Excuse the ignorance, but I learned for the first time today, I guess when Andy mentioned that one of the reasons eggs are so expensive is they use them for making vaccines. I didn't know that. Now, does a diseased chicken produce a diseased egg? Do the eggs have to get tested separately? Are they somehow immune themselves? Are these little compartments that are for genetic evolutionary reasons insulated from the mother bird being a carrier?
I believe so. I mean, I think the birds are infected, and so the flocks that produce the eggs, the layers that produce the eggs for normal inoculation for development of seasonal vaccines are affected. The eggs are not affected.
Yeah,
However,
not affected.
no,
So still
but-
could get some use out of them.
But there's a real problem. Yes, we can't get use out of any eggs that came from positive flocks.
Cool.
And also surrounding flocks. So today, for example, there's an example. I was in touch with Penag and I said, listen, the real reason we're not vaccinating is because it's complicated to do. It's expensive, but more importantly, like, you lose the export market. But there's another reason why the export of birds is restricted, and that is if you have influenza in the flock or a surrounding flock. So import countries will restrict trade with countries and states, subnational regions, if they have influenza. So Canada has now banned our eggs and chicks. So, it's a temporary ban, but what's worse, the chicken or the egg? Not to make a very bad joke, but the problem is the eggs themselves are not infected, but the flocks, and then it's now going to be a race to see whether the restricted export is more related to our influenza problem or the fact that we're introducing vaccines and waiting for their approvals. But it's a very good question. I'm not aware of virus transmission, of avian virus transmission to the eggs.
So this is kind of, I guess, theoretical. I'm not a scientist, but my understanding from way back of the nature of a virus is that it is not a living creature in the sense that it does not have the properties that we define, that we attribute to living creatures. So my question is it possible for you as scientists to imagine the ecosystem of this world in its totality without there being viruses at all? So in a kind of reshaping of that question, if I always ask myself, well, what the hell do viruses get out of it by reproducing so aggressively? And so if it were possible to find that which in the world of viruses caused them so single-mindedly to want to replicate, and that could be neutralized, would it be to our advantage to turn that desire off? Or are there too many beneficial viruses in the environment for that to make sense? I know it's complicated, but I can't help myself.
Well, I'll say that I'll say that what we know about beneficial viruses is much less than what we know about the pathogenic viruses, simply because we mostly worry about the viruses that make us sick. What drives viruses to just replicate? Viruses are obligate parasites, so they require a host to complete their life cycle, but they don't innately want anything. They're not thinking, living, breathing creatures with, sentient desires. They're simply nucleic acid wrapped in some protein that gets propagated along. So, and I will say that the diversity of viruses in the world is absolutely enormous, and so the viruses are part of every single ecosystem. They're part of our microbiome. They're part of ocean microbiomes. Viruses, phages are a particular type of virus that infect bacteria, and they're actually really important for bacterial recombination and the spread of genetic material between bacteria. So I think it's almost impossible to imagine a world without viruses and what that would look like. And it would probably shape a lot of things that we couldn't anticipate.
You have to ask the microbiome that question too, because as Louise said, there is an interdependence. But I would add one other thing ecologically, and I'm not an ecologist, but there is a certain amount of pressure on populations, wildlife populations, that relate to viral loads. And when they are forced to congregate in small areas, for example, they will sometimes exchange viruses in a way that they'll actually become immune to the viruses. And that's not necessarily a good thing. So there are some species that whereby it's thought that virus and virus selection, infection, and death of the animal, as there are in bacterial and parasitic infections, that eliminate subspecies and animals that may be weaker. So it's part of the natural selection process. So the unintended consequences of eliminating viruses might be, quite bad in that respect.
Okay, so first of all, do you think cage free or free range poultry is a more dangerous vector than those in cages because they might interact more with waterfowl? And I guess the other question is, do you have any thoughts on chronic wasting disease in deer?
I'll let Andy handle the chronic wasting disease question, but with regards to free range birds, there's definitely a known link between raising poultry outdoors and the frequency of infection with avian influenza virus, highly pathogenic avian influenza viruses. So we think that one of the main mechanisms by which these poultry species get infected with avian influenza is by direct interaction with these wild bird species. And some of these wild birds that are the natural reservoir for avian influenza can have these viruses and be totally asymptomatic. So especially ducks, we know that they can migrate really far distances and they can be shedding virus at pretty high levels without really being sick. So in environments where, there is opportunity for interaction between these wild and domestic bird species, we know that that does definitely increase the risk for infection.
That might be why the mortality of backyard birds is higher, because they're just much higher load of exposure. To answer the question, we're here to talk about pandemic potential right now. The prions that cause chronic wasting disease in deer and related diseases like spongiform encephalopathy in cattle and Creutzfeldt-Jakob disease in humans are not known to be zoonotic. However, there is the concern. And so we have at Penn actually a vast diagnostic network through the Wildlife Futures Program for testing deer that have been, killed by hunters for food consumption. And so they send them in and a week later they get the answer back. And I think there's a general sense not to consume deer that are positive for chronic wasting disease, even though they're asymptomatic and they look normal. But the zoonotic potential right now, if anything, is very low and would require probably a much longer longitudinal, much longer study to understand. But just for the purpose of safeguarding the human population, we do have that diagnostic paradigm in place. So it's a good question. Maybe a zoonotic disease on the horizon, but currently right now it's an unknown.
Okay, so I have three questions, but I'm going to try to combine it in two. And I have one specific question probably for Luis and then a question for the entire panel. You were very specific in saying that with regard to avian bird flu, that there were intrinsic immunity factors that probably were protecting humans and that the species were very different, obviously. And so I'm wondering then, is that a general principle that where the species are very different and then assumption of immune factors that would mean there's less likelihood of a animal to human jump? And so are there some species that we should be more worried about than others is the real question. So that's question number one to you specifically. And then a combined question to the entire panel that is both, what did we learn during COVID well and are applying, and what did we learn poorly and should be applying? And then to B or C would be, what should be the global coordination that you would ideally like to see to reduce the likelihood of zoonotic transmissions?
Yeah, so starting with the intrinsic host factors, and are there particular species that we should be more worried about than others? It's a really hard question to ask. It would make sense that we would tend to receive viruses from other animals that we are most similar to. But then again, we are not that similar to chickens, and so we still can get their viruses. So I would say that there's no hard and fast rule because zoonosis is complicated. It depends on how we're interacting with these species, as well as intrinsic innate immune factors, as well as the virus life cycle. So it's somewhat virus specific. That being said, there's very few zoonoses that I know of from like fish to humans. So really distant species, that's, I think, less common. The other question, what did we learn well from the pandemic and what should we do better? I think one of the things that there's a lot of stuff we learned from the pandemic. Something that I think we did really well is this genomic surveillance aspect that I've talked about. We sequenced so many genomes of SARS-CoV-2 and used them in really innovative and exciting ways to learn a lot about how these viruses are moving and spreading, and to help us, learn about variant emergence in real time. We learned about how great mRNA vaccines are. That's such an amazing innovation. And this is hopefully going to really change how we think about other viral vaccines. I think that we, all learned that masks work, which is something that's actually sort of new. We didn't have tons of data before this on how well masks work in large public health settings beyond kind of healthcare settings. And that's really great information to know. What would I like to see going forward? I think that something that perpetually comes up when we have outbreaks and then sort of fades away is investment in public health infrastructure just really takes you so far. You get so much bang for your buck by investing in public health. When people have access to testing resources and ways to find out if they're sick or not, and if they have the resources to really protect themselves and their families, we do a lot better. And when we think about infectious disease transmission as this population problem, instead of as individualized medicine, you can really get great benefits for the whole population. So I would love to see more investment in public health infrastructure that really prioritizes equity across large populations.
There's a number of lessons from COVID, and let's just keep in mind, very, very relevant question because it's a zoonosis. So we can look at it through the lens of SARS-CoV-2 being a zoonosis, just like MERS and just like the first SARS. And what did we learn in that regard in terms of like preventing the pandemic next time, which is the theme of this panel? We can also look at it from the perspective of a public health disease with the, as Louise said, the disparities that we witnessed and building out public health infrastructure. But I think to prevent the next pandemic, we have to first realize that these pathogens are global, right? And we have to understand, so we have to have a monitoring method, you have to have a detection method, means really robust diagnostic labs, and you have to have a reactive response method. Those are the three areas that anybody that's a student of public health will hear over and over and over again. And so what does that look like in the future in the context of preventing the next pandemic? It's important that we understand where transmission might happen. So you do need, rather than, and Louise and I have discussed this a lot, rather than having a global surveillance method where we're testing viruses from all over the world, sort of randomly, stochastically, and banking them and so forth, and putting them in freezers. And then we have the risk, as we all know, of freezer meltdown or some sort of a problem with the BL3 or BL4 facility leaking. We could go about this in a more targeted way where we look to see where people are interacting with animals, where people are going into caves, where people are interfacing with agricultural species next to deforestation, where people are interacting with ticks and so on and so forth. We go to the places where we know zoonotic transmission is highest risk. We go to those at the national and subnational level, and we make sure that the diagnostic surveillance capacity of those nations is good. Now, the problem is you have to have a lot of these substations of diagnostic work. And you also have to have culturally and socio-ecologically socio-ecological experts. You have to have people that understand and can talk to the local people about their habits, as we learned in many, many outbreaks that we learned in Nipah virus in Malaysia, that people were collecting palm juices and that the bats were peeing in the palm juice things, but no, nobody is supposed to, you're supposed to filter and pasteurize the palm juice, but people were taking a little bit away to make palm wine and not telling anybody, but it turned out they had to have local experts to interview people with knowledge, local knowledge, to kind of get that out. And they figured out that's how people were getting the Nipah virus, because it's easily transmitted from bats to humans through the palm juices. So you have to have that cultural, socio-ecological knowledge on the ground, the diagnostic surveillance going on in all of these areas, and in a targeted way. And then you have to have shared information across the globe. And that's something we do poorly. And that involves governance as well. And lastly, you then have to have a response. You have to have a public health response. How are you going to deal with this palm juice problem? You know, what's your response going to be to the movement of bats in years where they can't find food and they're congregating their agricultural areas? You have to have a response. So you have to have the monitoring method. And the monitoring isn't just monitoring viruses. From that example, you can see you have to develop models to predict, based on climatic conditions, based on ecological conditions, based on land use, where bats are going to move. And if they move, you have to have models to help you understand when those interactions are going to occur, what time of year, through what intermediate host, a reservoir host like a horse, and then you have to have a response, right? And the response in Ebola is almost like an exaggeration of the types of responses that we all learned from COVID, right? We have to have quarantine, and we have to have contact tracing, and we have to go back to the families, and Ebola was complicated because of its long incubation time. And so you have to have those response teams, and you have to have protocols in place. So you have to have all of that. But I do think a targeted way of getting at areas of risk, investing globally in those, sharing information through multiple organizations. And one other thing I'll say as a veterinarian, that we have to take an approach to all of this through a One Health lens, meaning that there's an inextricable link between animals, humans, and the environment and ecosystems. So whenever one of those three and the triad is affected, the other two will as well. And so when you're designing global surveillance, you should be bringing in, as has been done through the One Health expert panel, is the World Health Organization, the World Organization of Animal Health, the FAO, and the UN Environmental Program. And that's the quadrite organizations now that are working on these very response systems. Really important.
I just had a quick follow-up. So I was glad that you mentioned these four entities and the coordination that has to happen at the global level. But I wanted to kind of pick up on something you raised, each of you raised in different ways about issues of equity and this being perceived more as a public health issue and a population problem rather than an individual problem. And I kind of have a two-point question. So one is, what more global coordination is needed? Because clearly, Andy, the things you've just outlined seem very straightforward, but clearly aren't being done to the level and degree that they need to be. So what has to be like the push at the international level, and where should that type of public policy coordination happen? And then on the other side of that, this idea of approaching these issues as public health and population problems rather than individual problems, because so much of the way disease is dealt with, including this type of disease transmission, is very population or country focused. So you get Ebola and people are only thinking about West Africa until someone flies to the United States, just as a small example. So maybe a response to both those.
Well, I would say that only 95% of investments in the US goes into doctors and medicine and 5% into public health or less, right? So investments, and in the global level, this is World Banks and this is global financing, have to go into both addressing climate change as well as addressing public health in terms of this quadrate infrastructure. So investments are really key. And there's some really good programs, like a program called Stop Spillover out of Tufts that is working, is funded through the USAID to do just that kind of work, for example, as well as the agencies, the international agencies that I mentioned. So there have to be more investments in public health infrastructure. There's a very good book by Sandra Galea, the Contagion Next Time. I love the book because he says at least 100 times that we need to invest in the determinants of health. And so I think with that, let's just think about investing in the determinants of both animal health, human health, and ecosystem health, and do that in unison through the interagency relationships that we have and the knowledge that we've learned from COVID. And I think the solutions will be much more at hand. But we clearly need to invest in a sort of a public health and addressing the determinants of health globally, which also create the disparities of health as well, and would alleviate the disparities in health.
It's been striking me, I mean, beyond the sort of governance institutions over which we have limited control, individual scientists play a big role in sort of how we do or don't share that kind of information globally, right? I mean, I think about your research and like the relationships on which that kind of data sharing is dependent fall a lot on individual labs and scientists to sort of form those collaborations that help us get the samples we need around the world.
Yeah, I mean, that's definitely very, very true. But I will say another really great thing is that this seems to be really moving in a more open direction, I would say, in many areas of science, and especially people who work in global infectious diseases. There's a real movement towards being open with data and sharing it widely. And I think that increasingly I'm seeing more and more people who are really interested in collaborating and also really interested in giving proper scientific credit to people in other countries who are really doing like the challenging work. And I think that, you know, that really goes a long way in building up the trust to get better global data sharing.
Do we have time for one last question?
From an operations research standpoint, how could you minimize the lag between when you realize you have a new zoonotic disease and when you have a vaccine available for humans? I mean, would it make sense to take the likely suspects and develop all sorts of vaccines through animal testing at least, or perhaps through phase ones, so that, you know, you have something you could kind of get a jump start on, because there's a long I mean, it was amazingly shortened, but nevertheless, there is a long lag between when you have, you know, an epitope and when you have, you know, a vaccine that you can give to people.
Yeah, I think there's a couple ways to think about this. So the first case is for a fully new pathogen like SARS-CoV-2. I don't know how you would make that process faster while also guaranteeing that widespread deployment would be safe and effective. For viruses that we know cause spillover infections and cause disease in humans, we do have somewhat of a system to try to be prepared. So in the case of avian influenza, the World Health Organization actually has this vaccine strain selection process. It's the same as what they do for seasonal influenza viruses every year, but they also do it for avian influenza viruses. So they surveil across all of these different collaborating centers across the world. They say what viruses are circulating in your area, which viruses are you worried about? And then they actually pick those vaccine strains and they keep small bits or samples of those viruses in these freezers that these different labs maintain, such that if these viruses ever did go on to cause a pandemic or a larger outbreak, you could much more rapidly deploy these into a new vaccine. So for example, I was just reading today that the CDC just updated their H5N1 vaccine candidate to be the virus that's currently circulating in North America. And that is actually a very good match in terms of the sequence of the virus to what is currently circulating. So we do have a little bit of framework in place for that kind of thing.
I mean, I think you teed us up for a very optimistic ending because, in fact, lessons from COVID, we have learned vaccine development. We've learned a way of accelerating vaccine development from the technological platform stand, also to the surveillance to introduction of the vaccines, as well as the approval process. And so and when you're looking at what's available now, we have influenza vaccines for humans. So at present, we're not in big risk for this particular clade transmitting in its current form, and we're not able to vaccinate for it. There's a new Ebola vaccine, there's SARS vaccines, but I would just add one more layer to this, and that is, I think that rather than thinking about the reactivity of vaccination, whereas that's going to be very important, and in avian influenza, as Louise said, we take the information from the Southern Hemisphere 6 months in advance, and we turn that into a seasonal vaccine, and we expect sterile immunity. I don't expect to get even sick from influenza. I don't know if that's realistic. I think what we really want to do globally is keep people out of the hospitals and keep them alive. And so the universal vaccines that people are developing, as at Penn, for example, universal flu vaccine, they are not necessarily really efficient, and they contain 20 different, is strains the right word?
Yeah. Great.
Thank you. Subtypes of strains. I never get that right. Of the influenza vaccine, of influenza virus. So they're going to protect us from going to the hospital and from dying. And if we all had that vaccine, right, we would probably solve a major public health problem. So globally, I think it's going to be a layer of universal vaccines that are based on more elemental aspects, the stalk, for example, of viruses, and seasonal vaccines, which are more specific to the current extemporaneous strain, which will induce sterile immunity. In the end, for public health work, I'm not sure sterile immunity is going to be the luxury that we can all enjoy throughout history. But if we could keep people from dying, and if we didn't learn that from COVID, we've learned nothing. So
that mRNA vaccine research is really promising and extraordinary what's happening here at Penn in that vein. Thank you all so much. I think we're going to hear some closing remarks.
Join me in thanking our panelists. And thank you all for coming. The semester is winding down, so we have fewer and fewer events. But next week on Monday, April 24th at 4:30, we're going to have our final joint Prairie World House Andrea Mitchell Center for the Study of Democracy lecture series on democracy and emergent technology. That'll feature the Estonian ambassador to the United States, Christian Prick. As always, you can access recordings of programs like this on our YouTube channel. This will be up in a few days. Please join our mailing list, follow us on all our social media channels, and we have a grab and go snack in the student lounge in the back. So thank you all for coming, and please join me again in thanking our panelists.